Bioinformatics plays a crucial role in phylogenetic analysis and the recovery of the evolutionary history of vectors. Vectors, in this context, refer to organisms that transmit pathogens between hosts, such as mosquitoes transmitting malaria or ticks transmitting Lyme disease. The field of bioinformatics combines biology, computer science, and statistics to handle large-scale biological data and extract valuable information from it. This account will explore how bioinformatics has revolutionized phylogenetic analysis and our understanding of vector-borne diseases. Phylogenetic analysis is the study of evolutionary relationships among organisms based on their genetic sequences. It allows researchers to reconstruct the tree of life, which showcases the relatedness and divergence of various species over time. In the case of vectors, understanding their evolutionary history is essential in tackling vector-borne diseases as it can shed light on their origins, diversification, and adaptation to different environments. One of the primary applications of bioinformatics in phylogenetic analysis is the use of molecular data. Advances in DNA sequencing technologies have made it possible to obtain vast amounts of genetic information from various organisms, including vectors. Bioinformatics tools are then used to process and analyze this data, extracting relevant genetic markers for phylogenetic reconstruction. Initially, phylogenetic analysis relied on a single gene or a few genes to infer evolutionary relationships. However, with the advent of high-throughput sequencing, entire genomes can now be sequenced and used in phylogenetic studies. This has significantly improved the accuracy and resolution of phylogenetic trees, enabling researchers to understand vector evolution in greater detail. To construct phylogenetic trees, bioinformaticians utilize various algorithms, such as maximum likelihood and Bayesian methods, to infer the most likely evolutionary history based on the genetic data. These methods consider the rates of genetic changes over time and model the substitution patterns to estimate the relationships between different species or strains of vectors accurately. Furthermore, bioinformatics tools can handle large datasets, incorporating genetic information from a diverse range of vectors across different geographic locations. This enables researchers to study the global spread and diversification of vector-borne diseases, helping to identify the sources of outbreaks and understand how these diseases evolve and adapt to new environments. In addition to molecular data, bioinformatics also incorporates environmental data into phylogenetic analysis. This approach, known as phylogeography, combines genetic information with geographic data to understand the spatial distribution and movement of vectors and pathogens. By integrating data on vector movements with genetic divergence, researchers can trace the historical pathways of vector-borne disease transmission. Bioinformatics also facilitates the identification of potential drug and vaccine targets within the vectors and pathogens. By analyzing the genetic data, researchers can pinpoint specific genes or proteins that are crucial for the survival and transmission of the vectors and the pathogens they carry. This knowledge can lead to the development of targeted interventions to control the spread of vector-borne diseases. Moreover, bioinformatics has accelerated the study of vector-borne disease evolution by providing open-access databases and tools for researchers worldwide. Publicly available databases, such as GenBank and VectorBase, store and curate genetic sequences from various vectors and pathogens, making it easier for researchers to access and analyze relevant data. The application of bioinformatics in phylogenetic analysis has also led to the discovery of new vector species and disease variants. By comparing genetic sequences, researchers have identified previously unknown vectors and variations in pathogen strains, providing valuable insights into the potential for disease emergence and spillover to new hosts. In summary, bioinformatics has significantly advanced phylogenetic analysis and our understanding of the evolutionary history of vectors and vector-borne diseases. The integration of molecular data, environmental data, and advanced computational algorithms has allowed researchers to construct accurate and detailed phylogenetic trees, unraveling the complex relationships between vectors and the pathogens they carry. This knowledge is essential in designing effective strategies to control vector-borne diseases, mitigate outbreaks, and improve public health worldwide. As technology and bioinformatics continue to advance, our understanding of vector evolution and its implications